A very good evening to one and all who have joined us today through YouTube and various platforms. Good evening, everyone, and we welcome you all to the Medusane Forum. It gives me immense pleasure to introduce Medusane, a platform that has rapidly grown to become a trusted educational partner for medical students across India. Our mission is simple: to provide accessible and high quality education resources that empower students to excel in their academic and professional journey. With a vibrant community of over 23,000 YouTube subscribers, Medusane has made a significant impact in areas like clinical case presentation, theory lectures, MCQ discussions, career guidance, skill development, and so on. One of the things that makes Medusane stand out is our commitment to mentorship. While there are numerous platforms offering medical education, Medusane uniquely emphasizes personalized mentorship. We aim to provide students with daily guidance, one-to-one support, and practical insights that go beyond textbooks, helping them build confidence and clarity on their career paths. So, as we approach the World Diabetes Day in this month, we are reminded about the importance of awareness, education, and the collective effort in combating one of the world's most pressing health challenges, diabetes. This year, we are excited to present a special series of lectures dedicated to deepening our understanding on diabetics from its physiological biochemical foundation to its management and complication. So the World Diabetes Day is more than a date on the calendar. It's a global call to action. Diabetes affects millions of lives, impacting not only patients but families and communities. Through this series, our goal is to bring together experts in the field to share valuable insights that will empower healthcare professionals, students, and the broader community to tackle diabetes more effectively in their daily practice. So each session in this upcoming days is designed to address crucial topics from basics of insulin function to advanced management strategies, along with the latest guidelines. So the initiative emphasizes the role of continuous learning and collaboration in addressing diabetes and aims to equip you with practical knowledge and skills that are essential for improving patient care. To kickstart this series of lectures, we have our most respected faculties here. Before that, I call upon our own mentor of Medusane, HOD and Professor of Pathology, Dr. Jamna Rani ma'am, to address our meeting.
So good evening, one and all. We bring greetings from Medusane, and so this is our unique initiative on this World Diabetes Day 2024. And as all of you are aware, the theme for this year is breaking barriers and bridging gaps. So what we have decided is we'll bring a integrated series on diabetes, starting from biochemistry till the advanced aspect. So we will be covering all the topics in diabetes. So it will be an integrated learning series. And today we'll be dealing with biochemistry, tomorrow pathology, and we will move on. Even specialty-wise topics pertaining to diabetes will be there. So on one go, if you want to learn about diabetes in an integrated fashion, you can definitely follow this series. It will be of immense help for you in your university examination as well as in your day-to-day practice. So I really congratulate our team, Raghu and Parthiban, for this wonderful initiative. And today we have an excellent speaker, Dr. Jagdish, a passionate professor of biochemistry. So with his immense knowledge, I'm pretty sure this first day session of diabetes will be a very grand success. Thank you very much for the opportunity, and thank you guys. Wishing you all very best.
Thank you so much, ma'am. So before going to the session, I'd like to introduce our speaker of the day. So he's none other than Dr. Jagdish sir. He's an associate professor, Department of Biochemistry, Velamal Medical College and Research Institute from Madurai. He has awarded with the President's NB Gold Medal and Diplomate of National Board of Examinations. He has addressed national and international forums, published numerous articles and papers in national and international journals. He has received the gold medal for the best research paper in 21st National Conference of Ambicon held in Rohtak. So it is a privilege in having you here, sir. The session is all yours now.
Good evening, everybody. Thanks for having me here in this wonderful integrated session on diabetes, especially on to commemorate the World Diabetes Day program. So, as Dr. Jamna Rani rightly said, this whole series of lectures or seminars will help you to get the holistic picture of diabetes, starting from basic sciences to the clinical sciences. And I thank Dr. Raghunandan and his team for choosing me to give one seminar on this series of lectures. Hope I will give a better picture about the topic which has been assigned to me. So let me share my screen. Is my screen visible?
Yes, sir.
Yes. Okay. So the topic which I will be covering for the next 30 to 40 minutes is insulin, the hormone of life, unraveling its physiology and biochemistry. So, so I will start this session with this statistic about diabetes in India. In India, there's a study which was published in Lancet in 2023, which showed nearly 101 million people in India are living with diabetes, which is roughly 11.4 percentage. So if you see clearly here in this slide, it is more prevalent in the urban areas compared to the rural areas. So the prevalence of diabetes is constantly increasing constantly increasing in India, which is a matter of huge concern. So diabetes mellitus is actually a group of metabolic disorders which share a common phenotype called hyperglycemia. So diabetes mellitus can be caused due to two reasons. One, it can be an absolute insulin deficiency, there is no insulin at all, or it can be due to relative deficiency of insulin. That is, insulin is there, but insulin is not able to act. It's called as insulin resistance. These are the two major reasons for diabetes mellitus. And this hyperglycemia is a hallmark features of diabetes, which leads to various metabolic complications. In general, diabetes mellitus can be classified into four types: type 1, type 2, GDM, which is also called as gestational diabetes mellitus, that is diabetes mellitus diagnosed for the first time during pregnancy, and there are some other types. So, the one thing which keeps coming repeatedly is hyperglycemia, that is increased levels of glucose in the blood. So, which or how the blood glucose levels are maintained in the body? Hormones play a very important role to maintain the blood glucose levels within a narrow range. There are only two hormones which plays a vital role. One is insulin, one is glucagon. They play a central role in the maintenance of blood glucose. There are other hormones also that comes into play, which includes epinephrine, that is adrenaline, growth hormone, cortisol, and thyroid hormone. So these hormones will try to regulate blood glucose levels in your body based upon the physiological demands. So there's only one hormone which is responsible for lowering the blood glucose level in the blood, which is also called as a hypoglycemic hormone, which is nothing but insulin. Insulin is the only hormone which is responsible for lowering the glucose in your blood. That's what it's called as hypoglycemic hormone. Whereas the other hormones which I listed out in the earlier slide are responsible for increasing the glucose levels in the circulation. They are called as hyperglycemic hormones. One of the important hormones is glucagon, followed by epinephrine, cortisol, thyroxine, growth hormone. That's why these hormones are called as counter regulatory hormones. That is, these hormones counter the actions of insulin. That is, these hormones will try to increase the blood glucose because the insulin will try to decrease the blood glucose. So, in the scope of this lecture or this seminar is to focus on insulin. So we will cover about insulin. So why do we or how we regulate blood glucose level? This slide will help you to understand that. So we eat food. Once you eat food, in the blood, glucose levels will increase, and this will stimulate pancreas. A pancreas consists of alpha cells and beta cells, and this will stimulate beta cells of pancreas to produce insulin. And this insulin will try to push the glucose from the blood into the tissues. That is called glucose uptake. into the tissues like adipose tissue and skeletal muscle. So the glucose levels in the blood will drop, and glucose will get converted to glycogen, get stored in the liver and skeletal muscles. So that's how immediately after the food, the glucose spike in the blood is prevented by insulin. Whereas vice versa, when you're not taking any food or in the period in between the two meals, what happens? Glucose levels will drop in the blood. This will stimulate pancreas, and this will stimulate one release of hormone from alpha cells called glucagon. And this glucagon will try to release glucose into the blood by breaking down the glycogen which is already stored in the liver, or by increasing the production of glucose in the liver. Thereby, glucose levels will be restored. So this is how, in the normal homeostasis, glucose levels are maintained by insulin and glucagon. So we have to maintain glucose levels within this narrow limits. Okay. When I say narrow limits, what are those limits? So when you measure glucose levels in the blood, okay, after a fasting, you take food by 8 o'clock in the night. Don't take any food for next 8 to 10 hours. 0 calorie intake. And you take blood sample the next day morning in fasting. Your glucose level should be within 70 to 100 milligram per deciliter. So that is called normal fasting blood glucose. Or after a meal, take a breakfast and check your blood glucose. It should be less than 140 milligram per deciliter. So the hormones will play a role to maintain the glucose levels within these limits. Okay, especially insulin and glucagon. So today we are going to talk about insulin. Okay, fine. So how is insulin released from the pancreatic beta cells in response to an increase in blood glucose level? So I said when you take food, glucose will spike and that will release insulin. How does that happen? So what is the molecular mechanism behind that? So insulin is secreted from pancreatic beta cells. So glucose levels in the blood will increase immediately after a meal. So this glucose has to enter the pancreatic beta cell. The pancreatic beta cell is the one which stores insulin. Okay. So the glucose will enter into the pancreatic beta cell through this transporter called glucose transporter 1 or 2. Okay. There are two types. There are many isoforms of glucose transporters. One that is found in the beta cells are either glucose transporter 1 or glucose transporter 2. Through this transporter, the glucose will enter into the pancreatic beta cell. Once it enters into the pancreatic beta cell, glucose will undergo a metabolic pathway called glycolysis. Okay, that is, glucose will break down to form pyruvate. During this process, when the glucose breaks down to form pyruvate by glycolysis, two molecules of ATP is produced. Okay, and this pyruvate will enter into TCA cycle, and this TCA cycle will produce a lot of ATPs. Okay, and these ATPs, when the ATP levels increase in the beta cells, this ATP will close the potassium channel. There's a special type of potassium channel that you find in the beta cells. This is called ATP sensitive potassium channels. When the ATP levels increase, this potassium channels will close. When the potassium channel closes, it will lead to depolarization of the beta cell membrane. When there is depolarization, one more channel will open. That is called voltage gated calcium channel. The calcium channel will open, calcium will enter into the beta cell, and this will trigger exocytosis of insulin. Insulin will go out of the beta cell into the blood circulation. This is the exact mechanism how glucose stimulates release of insulin from a beta cell. Okay, so glucose undergoes glycolysis, what you see here, to form ATPs, and this ATPs will be increased in the blood in the beta cell, and that will close a channel called ATP sensitive potassium channel, which will cause depolarization of the membrane, which will open up the voltage gated calcium channel, and that will trigger exocytosis of insulin. So that's how insulin is released into the blood circulation with response to glucose. Fine. So there's one more important point regarding this insulin secretion is the insulin secretion is biphasic. Okay, that means the first phase is a dynamic first phase. It's a rapid phase, sudden peak in insulin levels, followed by a sustained steady state release. Okay, so this is very, very important because if you see here, this is what I explained to you in the previous slide, how glucose stimulates release of insulin. Okay, insulin is actually stored in the beta cells as granules, okay, in the vesicles. Okay, there are few vesicles just almost present on the near the membrane, like what you see here. That is called as RRP. RRP is nothing but readily releasable pool of granules. Those vesicles are staying close to the membrane Okay, and there are few granules which are away from the membrane. So when there's a glucose stimulus, the first set of granules that will get released is this RRP granules, readily releasable pool of granules, which will be released immediately. That's why you get a first phase, which is a dynamic phase. Followed by that, the other granules will be recruited to the membrane, and that will be exocytosed. That is the reason for the sustained steady state second phase. Okay, so this is how insulin is secreted from the beta cell in two phases. That is called the biphasic insulin secretion. First phase is caused due to the release of readily releasable pool of granules, which is present close to the membrane of the beta cell. The second phase is because of the reserve pool, which is present far away from the membrane. So that will come from there to the membrane and then you get exocytosed. Okay. So this is what is how insulin is secreted from the beta cells in two phases. Fine. So next important point is the insulin is actually secreted as a pro-insulin form. Okay. So like how we have studied like pro-proteins is converted to proteins. For example, the best example is trypsinogen is converted to trypsin. Chymotrypsin is converted to chymotrypsin. Okay, pepsinogen is converted to pepsin. Like that, insulin is also secreted as proinsulin. It is an inactive form. And this proinsulin is made up of, this is the structure of the proinsulin molecule. Okay, as you can see, there's a single amino acid chain. Okay, and there are two disulfide bonds. Like the disulfide bonds helps the protein to get folded. Okay, and this proinsulin will be processed to form two molecules. One is a mature insulin molecule, which is released by the beta cell, and there's one more molecule called C peptide, which is also called as connecting peptide. Okay, so what you see is a connecting peptide here. If you follow my arrow, you can see this is a C peptide, which connects this two chains of insulin molecule. Okay, so insulin is made up of two chains, A chain and B chain. A chain is 21 amino acids in length, B chain is 30 amino acids in length. So in general, insulin is made up of 51 amino acids. Okay, so this is the structure of insulin. So this is what I told you. This is a C peptide. And this green part is a mature insulin molecule, which consists of A chain and B chain connected by disulfide bridges. Okay, fine. So why I am telling you about this is when insulin is released from the pancreatic beta cell, C peptide is also released. So there's C peptide and insulin are stored in the secretory vesicles and are both released in equimolar concentration, almost the ratio of 1:1. Okay, so why I should know about this? This is because if you want to estimate beta cell secretory capacity or beta cell reserve of pancreas, you can check C peptide levels, which will give you a clue about the beta cell secretory reserve. You can ask me why can't we measure directly insulin? Okay, we can measure insulin, but insulin's half-life is relatively short when compared to C peptide. Okay, so in general, human pancreas will secrete about 40 to 50 units of insulin daily. And this represents about 15 to 20% of the hormones which are stored in the beta cells. So as I told you earlier, the insulin's half-life is 5 to 10 minutes. And C-peptide's half-life is 30 to 35 minutes. It's relatively longer than insulin. Okay, that's why estimating C-peptide in the blood will give you a better index of insulin secretory reserve or beta cell secretory reserve. Okay, fine. So these, we can measure insulin in the blood, C-peptide in the blood, which are all the tests which we do to assess the functions of pancreas, okay, especially in relation to diabetes. Right. So now we know how insulin is released from the beta cell. We know the molecular mechanism behind the release of beta cells. Now we are going to see how it acts. What are various actions of insulin? It is, we know the overall action of insulin. The overall action of insulin is to decrease blood glucose level because it's hypoglycemic hormone. So insulin acts on these target tissues, especially the skeletal muscle and the adipose tissue, and the skeletal muscle. So insulin acts on these cells by interacting with its receptor called insulin receptor. Okay. Insulin comes and binds to this insulin receptor, and this triggers or this activates the receptor, and this triggers multiple signaling pathways. It activates multiple signaling pathways, which ultimately causes activation of few enzymes called as kinases. Kinases means that will add a phosphate group and will activate phosphatases. Phosphatases means remove the phosphate group. Thereby, it inhibits certain pathways and activates certain pathways. Second, it causes translocation of a transporter called GLUT4. Okay, we will discuss about this in the subsequent slides. Third, it is responsible for cell growth. Okay, so this is how insulin. This is the overview of how insulin acts. Insulin acts by binding to its receptor called insulin receptor on the target cells called skeletal muscles and adipose tissue, and activates various signaling pathways leading to all these metabolic effects. We'll see one by one. Okay, first, but the first thing about the insulin receptor is the insulin receptor is slightly unique. Okay, insulin receptor has an intrinsic tyrosine kinase activity. Okay, and it is a tetramer. It consists of 2 alpha chains and 2 subunits, 4 subunits, 2 alpha subunits, and 2 beta subunits. So this is an insulin receptor, which is a free receptor. As you can see, this is just a receptor. And what is this? Why is those tyrosine residues? See, receptors are nothing but proteins. Proteins are nothing but made up of amino acids. One of the amino acids which we are going to deal here is tyrosine. So there is tyrosine molecule present in this receptor. So this is an insulin receptor which is free. Once this insulin receptor binds the insulin, what happens is this tyrosine or this receptor itself has tyrosine kinase activity. I said kinase is nothing but it adds a phosphate group, right? So once insulin comes and binds to this receptor, it activates the tyrosine kinase function, and thereby the tyrosine residues will get phosphorylated. Okay, so this P is responsible. P indicates phosphorylation of the tyrosine residues. Once this happens, it activates a cascade of phosphorylation. Okay, that's called phosphorylation cascade. Okay, so insulin binds to the receptor, tyrosine kinase is activated, tyrosine gets phosphorylated. Once this happens, there is a series of phosphorylation happening in the target cell, which is nothing but the skeletal muscle or the adipose tissue. Okay, now let's see little bit in depth. What is that phosphorylation cascade? Okay, so insulin acts on this insulin receptor. I said tyrosine gets phosphorylated. The next subsequent is it phosphorylates a substance called insulin receptor substrate. It's called IRS, which gets phosphorylated, and that will phosphorylate another 2 enzymes called PA3 kinase and PDK1 that ultimately culminate in activating a protein called AKT, which causes GLUT4 translocation. Okay, so this is how insulin stimulates translocation of GLUT4. We'll come to it. What is GLUT4 translocation? We'll come to it in the subsequent slide. Then it also activates another kinase called P70S6 kinase, which is responsible for regulating the enzymes of various pathways. Then it activates an important pathway called mTOR pathway, mammalian target of rapamycin pathway, which is responsible for regulating the gene transcription. That's how insulin regulates the gene transcription of certain enzymes. That's how insulin regulates gene transcription of many proteins. Then they're also responsible for cell growth by activating this pathway called GRB2 mSOS pathway. So this is the molecular mechanism of insulin signaling. Okay, so insulin binds to the insulin receptor and triggers the phosphorylation cascade, which is responsible for various end target functions of insulin. Okay, which include GLUT4 translocation, enzyme activity, gene transcription, and cell growth. Right. First, as this keeps coming, translocation of GLUT4, what it is? See, GLUT4 is a glucose transporter. This is present in 3 tissues: adipose tissue, heart, and skeletal muscles. These 3 tissues will take up glucose only in the presence of insulin. Okay, so GLUT4 is an insulin-dependent glucose transporter. That is, for example, in the absence of insulin. So when the insulin levels will be absent in your body and the blood glucose levels will drop. Okay, in the absence of insulin, GLUT4, which is present inside these cells, that is skeletal muscle cells or adipose tissue cell, will remain within the cell. It won't go to the surface. So what does it mean? It doesn't go to the surface. If it doesn't go to the surface, glucose cannot come into the cell. So glucose will stay in the blood itself. That's what we need, right? Because there is no glucose in the blood. So you don't want to take up glucose and use it up. Okay, you preserve it for the serious energy demands. So the GLUT4 will not go to the surface. Okay, so what happens in the presence of insulin? In the presence of insulin, insulin will act on insulin receptor, causes this GLUT4 translocation to the membrane. So the GLUT4 will go to the membrane, and that will be responsible for taking the glucose from the blood into the cells. Okay, so this is a very important function of insulin. That's why they say insulin stimulates peripheral glucose uptake. So if there is no insulin in your body, or the insulin is not able to act, which is what happens in diabetes mellitus. Glucose cannot come from the blood into the cell, so glucose will stay back in the blood itself. That is the reason why patients with diabetes mellitus have hyperglycemia. Okay, because only in the presence of insulin, GLUT4 can go to the surface. Okay, if the GLUT4 cannot go to the surface, okay, glucose cannot be taken up into the cell. The peripheral glucose uptake is blocked, so glucose will stay back only in the blood. That is the reason for hyperglycemia. Okay, fine. The next important function is, as I told you earlier, insulin regulates various enzymes. Okay, how does insulin regulate various enzymes? I am not going into detail of each and every enzyme here, but the one simple thing of it that which you want to remember is hormones regulate enzymes by covalent modifications. That is, they can either activate them or inhibit them by causing either phosphorylation or dephosphorylation. That's it. Okay. So insulin activates certain enzymes and insulin inhibits certain enzymes of various metabolic pathways. How does it do it? It just causes either phosphorylate that enzyme by stimulating a kinase, or dephosphorylate that enzyme by stimulating a phosphatase. That's it. Okay. So I don't want to go into detail of each and every enzyme because that is going to be confusing. Okay, fine. So having we saw how insulin is secreted, we saw how insulin is how insulin signal is happening. Now we are going to see the effects. Okay, what are the effects of insulin on glucose metabolism? What is the effects of insulin on lipid metabolism? What is the effects of insulin on protein metabolism? Okay, fine. So insulin stimulates glucose uptake, I just now told you, by translocating GLUT4 to the peripheral tissue membrane, cell membrane, adipose tissue, heart, and skeletal muscle. Insulin will promote glycolysis. That means insulin will stimulate the breakdown of glucose to form pyruvate. Okay. Insulin stimulates this enzyme called glucokinase, which is also a glycolytic enzyme. Insulin stimulates glycogenesis. That means glucose is stored in our body in the form of glycogen, especially in the liver and the skeletal muscle. Okay. Insulin promotes this. Insulin promotes conversion of glucose to glycogen. So if you see carefully, it promotes glucose uptake, it promotes breakdown of glucose, it promotes conversion of glucose to glycogen. Okay, that means it does everything to reduce blood glucose level. Okay, at the same time, it inhibits glycogenolysis. Insulin inhibits glycogen, that is, insulin prevents breakdown of glycogen to form glucose. Insulin prevents gluconeogenesis. Insulin prevents synthesis of glucose in the liver. So ultimate overall effect is insulin decreases the glucose level. Okay, so that's it. Because it promotes glucose utilization and glucose uptake, and it inhibits glucose production. Thereby, it brings down the blood glucose level. So this is the overall effect of insulin on glucose metabolism. Okay, when I told you in the earlier slide, it may not have made much sense. Having discussed the insulin signaling and its action, now you will be able to correlate how insulin decreases the blood glucose level. Now the effect of insulin on lipid metabolism. Okay, see, insulin is actually a lipogenic hormone. What do you mean by lipogenic hormone? Insulin will convert glucose to form acetyl-CoA through glycolysis, and pyruvate will be converted to acetyl-CoA. And this acetyl-CoA will be used for the synthesis of fatty acids, which will be used for the synthesis of triacylglycerol, which is the storage form of fat in our body. Okay, so insulin stimulates lipogenesis. So insulin stimulates the synthesis of triacylglycerol. Insulin inhibits lipolysis. Insulin will not allow the fat stored in the adipose tissue to break down. Okay, so insulin is actually a lipogenic hormone. Okay, so this is insulin is a lipogenic hormone. It has an anti-lipolytic effect. Okay, this is very important because when we discuss the metabolic derangements in diabetes, this is going to help you in understanding that. Okay, which we'll see in subsequent slides. So that's about the effect of insulin on lipid metabolism. Effects of insulin on protein metabolism. it has only one effect. Okay, it stimulates protein synthesis. In a way, insulin is more of anabolic in nature. So insulin stimulates protein synthesis. Okay, so to summarize the effects, it decreases blood glucose. It increases the synthesis of triacylglycerol. It's like that is, it stimulates lipogenesis. It stimulates protein synthesis. Okay, so now we will coming back to the original picture. Diabetes mellitus, I said it's a group of disorders which share the common phenotype of hyperglycemia. I said we can classify diabetes into 4 types: type 1, type 2, type 3, type 1, type 2, GDM, and other types. Okay. So what happens in type 1 is there is autoimmune destruction of beta cells. So beta cells is not at all there. Okay. So insulin is not at all produced. That causes absolute insulin deficiency. First point. Okay. What happens in type 2 diabetes? Type 2 diabetes, insulin is produced, but this insulin is not able to act on the target cells. What are the target cells? We just saw adipose tissue, skeletal muscle, and other cells. So insulin is not able to act. This is called insulin resistance. So in order to overcome this insulin resistance, beta cells will produce a lot of insulin. So the initial stage of type 2 diabetes, you can see patients with fasting hyperinsulinemia. That's because of this fact. To overcome insulin resistance, beta cells will overfunction and produce a lot of insulin to overcome this resistance. Okay, so that's why we see that the initial stage we will see a lot of patients with fasting hyperinsulinemia. Okay, fine. So this is about type 2. Type 1, absolute insulin deficiency. Type 2, this insulin resistance. Okay, so but this compensatory overfunctioning of beta cells to produce a lot of insulin will not be there for will try to do this only for a certain period of time. After that, beta cells will undergo dysfunction and they will go on to apoptosis. Type 2 patients also will develop beta cell secretory defect. Okay. So what I am trying to tell is it will try to overcome the resistance by producing a lot of insulin, but only to a certain point of time. After a certain point of time, it cannot keep on producing insulin. So it will also go for apoptosis. So type 2 patients will eventually end up in beta cell secretory defect. Okay, fine. So this is a natural history of type 2 diabetes. Okay, there are 3 curves here. Okay, the first curve is insulin resistance. As you see, it is slowly increasing. Okay, the second curve, the yellow curve is insulin levels. As insulin resistance develops, your beta cells will try to produce a lot of insulin. That's what I said, hyperinsulinemia, but only to a certain point of time. After this, beta cells will undergo apoptosis, the insulin levels will drop. Okay. The third curve is a glucose curve. As insulin resistance develops, glucose levels will increase, leading to hyperglycemia. Okay. This in-between stage is called pre-diabetes. Okay, which is of matter of concern here because almost all patients with type 2 diabetes will go through this pre-diabetic stage. Okay. Even people with pre-diabetes are at increased risk of heart attack and stroke. Okay. So pre-diabetes stage, if you catch any patients in that pre-diabetic stage, we should intervene. We should give them dietary advice. We should give them lifestyle modification so that we can delay their progression into type 2 diabetes. Okay. So that is, this figure tells you about what is pre-diabetes and what is insulin resistance and so on and so on. Okay. So whether it is type 1 or type 2, it is either insulin deficiency or insulin resistance. Okay. So what happens if there is insulin deficiency or insulin resistance? There is decreased glucose uptake. Okay. Insulin inhibits glycogenolysis. Since insulin is not there, it is no longer inhibited, so glycogen will break down. Insulin inhibits gluconeogenesis. Since insulin is not there, gluconeogenesis will be increased. All this will lead to hyperglycemia. Okay, I am just putting everything in one picture now. Okay, so that is the reason for hyperglycemia in patients with diabetes, whether it could be type 1 or type 2. Insulin, as I told, promotes protein synthesis. So when there is no insulin, proteins will break down in your body. Okay, and protein breakdown will lead to will produce some substrates which will be eventually used for producing glucose. That is called gluconeogenesis. That will also contribute to hyperglycemia. One. As I told you earlier, insulin is a anti-lipolytic hormone. So when insulin is not there, there will be increased level. There is increased lipolysis, increased breakdown of triacylglycerol in the adipose tissue. That will release a lot of free fatty acids. And these free fatty acids just will alter the metabolism of lipoproteins in your body, which are responsible for carrying cholesterol and triglycerides, leading to dyslipidemia. And there will be a lot of free fatty acids in the blood, which can cause lipotoxicity, which can itself trigger inflammatory state. Okay, free fatty acids can trigger a pro-inflammatory state. Okay, so this is what happens in patients with diabetes mellitus because of insulin deficiency resistance. Okay, so insulin, if it's not there, we'll develop hyperglycemia, we'll develop dyslipidemia, there will be increased levels of free fatty acids in the blood circulation. Okay, so having said, I almost covered 90%. I'll just conclude with this two more slides. Okay, so if you see classical clinical pictures of diabetes, okay, these are the classical polyuria, polydipsia, polyphagia, weakness, and fatigue. Okay, so why do patients develop polyuria? Why do patients with diabetes mellitus develop polyuria? Okay, I am bringing this here because this will make more sense because we just now saw what is hyperglycemia, why there is hyperglycemia in patients with diabetes mellitus. Okay, see, kidneys will try to reabsorb glucose till the blood glucose levels are around 180 milligram per deciliter. That is called renal threshold for glucose. Okay, when the blood glucose reaches or crosses more than 180, kidneys cannot reabsorb glucose. So that's the reabsorptive capacity of kidney. When when the glucose levels exceed this reabsorptive capacity, glucose will come out in the urine. Okay. When the glucose comes out in the urine, glucose is an osmotically active substance. Okay, that will cause osmotic diuresis. Okay, so there will be increased frequency of urination. Because of this, there will be polydipsia. Okay, there's increased in thirst. Okay, even though there is a lot of glucose, these glucose levels cannot be utilized because there is no insulin. So there will be polyphagia. Okay, so I want to conclude with this. Okay, so insulin is considered as a life hormone because it regulates blood glucose levels. It's the only hormone which is responsible for reducing the blood glucose levels in the body. Okay, fine. So when you take food, glucose levels increases. That will stimulate pancreatic beta cells to produce insulin, and the insulin will stimulate the glucose uptake by translocating GLUT4 to the membrane. So the glucose will enter into the adipose tissue, and glucose will enter into the skeletal muscle, and glucose will be converted to glycogen. So this will bring down the glucose levels in the body. Okay. So this is how insulin regulates blood glucose levels in the body, and we have covered how insulin acts and what are the metabolic effects of insulin. So thank you thank you Medusane, and thank you Dr. Raghunandan and his team. Thank you Dr. Jamna Rani for giving me this opportunity to share my thoughts on insulin, the hormone of life. Thank you.
Thank you so much, sir. It was really a wonderful session. So, on behalf of the Medusane as well as the organizing team of World Diabetes Day 2024 lecture series, it is my pleasure to thank you for such a wonderful start for this kickstart for this entire series. So, today we just begin with the very basic, and this session has surely shed light on the insulin's profound role in our body and the science behind all those regulating mechanisms, metabolism, and energy that is very well necessary for even a clinician or anyone for both prevention as well as management of diabetes. Hence, that's the reason why we started with this topic. I would like to thank Dr. Jagdish sir, Associate Professor of Biochemistry from Velamal Medical College, Madurai, for allotting his time in spite of his busy schedule to come here and share his knowledge for you all. So this series is starting today and it's ending on 21st. And there was lots of background work team which had curated this entire thing. So as a founder of this platform and as a senior for everyone, I would like to express my sincere thanks on this day one itself. And for sure, this session is going to be fun-filled as well as a knowledge-filled session. So thank you, Parthiban, especially who is like who has just prepared this set of lecture series and took an initiative to do this. I would like to ask Parthiban to conclude the session with his vote of thanks. Thank you, sir.
Thank you. Thank you.
Thank you, Na. So thank you so much, sir, for this wonderful session. It proved to be a great beginning to our Diabetes Day series, and we are expecting more and more of you in future in our medicine channel. Thank you once again, sir, and thank you, viewers, for tuning in. I think today we have one of the greatest turnout in viewership today, I suppose. Thank you for the great start, sir. So stay connected with us, all the viewers. Thank you once again. We'll meet soon tomorrow.
Thank you. Thank you.
On behalf of the team as well as our advisor, I would like to thank sir. With this certificate is precious for us to give you as a memorable sessions for giving us this memorable sessions. Thank you so much.
Thank you. Thank you.
Thank you, sir.